Hello, and welcome to my lesson on glycolysis, gluconeogenesis, and aldolase gene expression. I'm teacher Joseph, and I'll be covering the basics of these, as well as expanding upon last lesson's notes. Okay. So let us begin with our first one, glycolysis. Glycolysis is the major pathway for glucose oxidation, occurs in the cytosol of all cells, is unique in that it can function either aerobically or anaerobically, depending on the availability of oxygen and intact mitochondria. It allows tissues to survive in presence or absence of oxygen. For example, skeletal muscles and RBCs, which lack mitochondria, are completely reliant on glucose as their metabolic fuel and metabolizes it by anaerobic glycolysis. So as we can see in our picture over here, let's just take a quick little gander. This is the general process of glycolysis that we'll be going over in the next few minutes. Very nice. And of course, it goes from glucose, 6-phosphate, down to pyruvate and lactate. Okay, so another name for this is the Embeden-Meyerhof pathway. Glycolysis converts glucose, as said, to two molecules of pyruvic acid. Pyruvic acid is more oxidized than glucose, so the energy released from the oxidation is used to create two molecules of ATP from two molecules of ADP and two inorganic phosphates. This is an anaerobic process. Under anaerobic conditions, the pyruvic acid can be fermented to lactic acid or to ethanol plus carbon dioxide. Under aerobic conditions, glucose is oxidized all the way to carbon dioxide and water. Awesome. So now let's quickly go through the steps of glycolysis. First one being that the substrate glucose is phosphorylated by hexokinase, resulting in the product glucose 6-phosphate. The source of the phosphoryl group in ATP and the expenditure of ATP early in the pathway works as energy debt necessary to get the pathway started. Next, the product of step 1 is rearranged to the structural isomer fructose 6-phosphate by the enzyme phosphoglucose isomerase and converts an aldose to a ketose. We'll come back to aldose a little later on. Now we finished the first two steps, let's continue on through steps 3 through 10. Step 3 of glycolysis is when the substrate fructose 6-phosphate is phosphorylated by phosphofructokinase, resulting in the product fructose 1,6-bisphosphate, with the source of the phosphoryl group being ATP yet again. Next, the product of step 3 is split into two 3-carbon intermediates by the enzyme Aldolase, forming glyceraldehyde 3-phosphate, which is used in the next reaction, and dihydroxyacetone phosphate. The dihydroxyacetone phosphate is rearranged into a second glyceraldehyde 3-phosphate by the enzyme triose phosphate isomerase. So those are the first 3 of this slide. Okay, so let's just take a quick note of that. So this over here is 3 through 5. Now we're moving on to steps 6 through 10. Awesome. Step 6. That is when the substrate glyceraldehyde 3-phosphate is oxidized to a carboxylic acid by glyceraldehyde 3-phosphate dehydrogenase. This reaction reduces NAD+ to NADH and results in the product 1,3-bisphosphoglycerate, with the new phosphate group attached with a high energy bond. Next, the high energy phosphate group is transferred to ADP by phosphoglycerate kinase, resulting in 3 phosphoglycerate and ATP. This is the first substrate level phosphorylation of glycolysis. That takes us into step 8. So in step 8 of glycolysis, 3 phosphoglycerate is isomerized into 2 phosphoglycerate by the enzyme phosphoglycerate mutase, which moves the phosphate group from carbon 3 to carbon 2. Then the enzyme Aldolase catalyzes dehydration of 2 phosphoglycerate, creating phosphoenol pyruvate, which is energy rich and is the highest energy phosphorylated compound in metabolism. Finally, in step 10 of glycolysis, the final substrate level dehydration in the pathway of phosphoenol pyruvate serves as a donor of the phosphoryl group transferred to ADP by pyruvate kinase, making ATP and releasing water, with the final products being pyruvate, as well as 2 NADHs, and which is, of course, our net ADH, and a net gain of 2 ATP molecules. Perfect. Up to this point, we've gone over the steps of glycolysis and a little bit about what it is. Now let's dive a little bit deeper by doing a complementary process, which is called gluconeogenesis. In gluconeogenesis, glucose can be synthesized from non-carbohydrate precursors. The brain has a strong preference for glucose, while the red blood cells have an absolute requirement for glucose. So for example, the brain needs 120 grams of glucose per day. The liver has about 190 grams stored of glucose as glycogen, or about a day's supply. Glucose can be synthesized in the liver from pyruvate, glycerol, and amino acids. Gluconeogenesis is not the reverse of glucolysis, though. So 2 pyruvate plus 2 ATP plus 2 NADH, 2 hydrogen ions, and 2 molecules of water will then result in glucose, 2 ADP, 2 inorganic phosphates, and 2 NAD pluses. And continuing on thermodynamically, the delta G of that reaction would be approximately plus 20 kilocalories per mole. And when you talk about gluconeogenesis, you also need to consider the Cori cycle. And in the Cori cycle, lactate from skeletal muscle is transferred to the liver. It is converted to pyruvate and then glucose, which can be returned to the muscle. Wonderful. Before we get into aldolase, let's just quickly go over the connection between glycolysis and gluconeogenesis. So on the surface, it may seem like they're simply opposite processes and thus reversal of each other, but that's not quite true. There are 3 non-reversible steps of glycolysis that can be bypassed, starting with the pyruvate kinase reaction. And the 2 enzymes that catalyze this reaction are pyruvate carboxylase and PEP, carboxyl kinase. Pyruvate carboxylase catalyzes pyruvate bicarbonate and ATP in order to produce oxaloacetate, ADP, and inorganic phosphate. While the PEP, carboxyl kinase, catalyzes oxaloacetate and GTP to produce PEP, GDP, and carbon dioxide. Fructose 6-phosphate is formed from 1,6-bisphosphate by hydrolysis of the phosphate ester at carbon 1. Fructose 1,6-bisphosphate catalyzes the exergonic hydrolysis, and the last step has the formation of glucose by the hydrolysis of glucose 6-phosphate in a reaction catalyzed by glucose 6-phosphatase. Now on to aldolase. The first point I want to make is that aldolase is an enzyme that forms and cleaves carbon-to-carbon bonds, as well as converts glucose into energy. The main locations where you'll find this protein is in the kidneys, livers, intestines, very small part in the brain, nervous system, but mostly is found in muscle tissue. If aldolase is increased in your body, it could put it in physical harm, because greater than normal levels of aldolase may be due to damage of skeletal muscle, hepatitis, infectious mononucleosis, liver, pancreatic, prostate, or liver cancer, muscular dystrophy, among many others. As well as there's many different types of aldolases with different substrates and products. Most common of these is fructose 1,6-bisphosphate aldolase, which is also commonly called simply aldolase. Because it is so common, it is often referred to as aldolase, and all the other aldolases must be specified in order to avoid confusion. Like anything. Further elaboration. There are two classes of aldolases, which have different catalytic mechanisms. The first one being class 1 enzymes that are only found in plants and animals. They don't require metal ions. They are characterized by the formation of shift-base intermediates between an active lysine and a substrate carboxyl group, while the class 2 enzymes are produced in bacteria, protists, and fungi and require an active site divalent metal ion. Many species of plants and animals have both types of enzymes, but two different types of aldolases are completely unrelated. So they have different structures and sequences that, even though they catalyze the same reaction, isozymes are found in each class of enzyme, and in invertebrates, the genes encoding each of them show tissue-specific expression. The three that we'll be covering here are aldolase A, B, and C. For example, class 1 aldolase A is expressed in muscle, aldolase B in liver, kidney, stomach, and intestine, and aldolase C in brain, heart, and ovary. The different isozymes have different catalytic functions. Aldolases A and C are mainly involved in glycolysis, while aldolase B is involved in both glycolysis and gluconeogenesis, with hereditary fructose intolerance being a side effect of a defect within aldolase B. Each of these play an important role in giving the body the proper energy that is needed. Aldolase A is a glycolytic enzyme that catalyzes the cleavage of fructose 1,6-bisphosphate to glycerol 3-phosphate and dihydroxyacetone phosphate. A critical role for aldolase A in physiology is demonstrated by human diseases that result from mutations in the aldolase A gene. The only aldolase A mutations that have been discovered are relatively mild, leaving the enzyme activity relatively unchanged. But that being said, although there is a wide variety of aldolases, the majority of the studies have found that class 1, fructose 1,6-phosphate, aldolase of vertebrates, more particularly the enzyme isolated from rabbit muscle, aldolase A, class 1, fructose 1,6-phosphate, bisphosphate aldolase, was first crystallized by Wolberg and Christensen from rat muscle. Taylor and associates developed a simple procedure for the isolation of large amounts of aldolase from rabbit muscle that greatly aided in the further investigations of this enzyme, which ultimately led to rabbit aldolase A being the most extensively characterized class 1 aldolase. Rabbit aldolase A was the first aldolase for which an amino acid sequence was published. Demonstration of the rabbit aldolase A messenger RNA sequence by cDNA cloning conclusively demonstrated that rabbit aldolase A has 363 amino acids and corrected several errors in the published amino acid sequence. Now, as to the structure, the tertiary and quaternary structures of rabbit and human aldolase A has been determined through studies utilizing X-ray crystallography. And in that comparison, they found that the structures are relatively identical, but the um, carboxyl terminal regions of the polypeptide is believed to be mobile, perhaps moving in and out to the active site, such that the electron density of these amino acids is often disordered. Aldolase A, despite being the most extensively studied isozyme of the aldolase family, has a catalytic mechanism that is not yet completely understood. It is still unclear what roles are played by several key amino acid residues at the active site. The aldolase B isozyme possesses a critical role in fructose metabolism and gluconeogenesis. Mutations in the human aldolase B gene, which result in diminished aldolase B activity, are the causes of an autosomal recessive disease, hereditary fructose intolerance, or HFI for short, which leads to excessive levels of fructose 1-phosphate in tissues expressing aldolase B. Several hazardous metabolic effects result in liver and kidney damage. The symptoms of the disorder, such as abdominal pain, vomiting, and hypoglycemia, result from the ingestion of fructose or similar sugars that are metabolized through fructose 1-phosphate. This disease possesses the greatest risk to patients during weaning, during infancy, which could result in being quite lethal. Growth retardation, renal tubular dysfunction, liver failure, coma, and death may occur if the disease remains undiagnosed and the patient continues to ingest fructose. Furthermore, the catalytic efficiency ratio of gluconeogenesis to glycolysis, synthesis and cleavage to be precise, shows aldolase B is also the most efficient isozyme, having an 18-fold higher ratio of specificity over aldolase A and 3-fold increase over aldolase C. It appears that aldolase B is well suited for its dual roles of gluconeogenesis and the metabolism of fructose in the liver. The expression of aldolase B is restricted to the liver, kidney, cortex, and small intestines. However, the control mechanism of aldolase B expression is not yet known. Glucocorticoids are released upon hypoglycemic stress. These hormones control the transcription of other genes for gluconeogenesis, such as PEPCKs or phosphoenolpyruvate carboxyl kinases, which I discussed a little earlier. The description of aldolase B gene is itself stimulated by glucose and insulin. Furthermore, the messenger RNA levels of aldolase B have been shown to increase in experimental studies where rats are fed fructose. The promoter in aldolase B has several liver-specific cis elements, both within 200 base pairs upstream of the 5' start of transcription and an element in the first intron. The third vertebrate aldolase isozyme, aldolase C, is found in the brain and other tissues of neurological origin. This enzyme has catalytic properties intermediates to those of aldolases A and B. Although the specific physiological role of this enzyme is unknown, it has been suggested that aldolase C, like other isozymes such as hexokinase 1 and lactate dehydrogenase H, is found in tissues of constant metabolic activity. Recent evidence indicates the occurrence of aldolase C in some tissues that can substitute for aldolase B in fructose metabolism. Comparisons of amino acids and nucleotide sequences have shown that aldolase A and C are more closely related to each other than either is to aldolase B. In contrast, the kinetic difference between aldolases A and C are not sufficiently distinct to explain why both isozymes are expressed in the brain. This phenomenon could be explained by the subtle differences found during glycolysis in the neural cells that express aldolase C. Alternatively, minor structural differences could be essential for supramolecular associations, particularly in each isozyme. For example, aldolase A's actin binding property or aldolase C's S100 protein binding property. Although expression of aldolase C has been reported in Xenopus ovary, it is not clear what role the isozyme has in this tissue. Aldolase C is distributed heterogeneously in the mammalian brain associated with the brain S100 protein. Furthermore, aldolase C has been identified as zebrin-2, an intracellular antigen associated with the organization of mammalian cerebellum, which structures itself in bands with sets of cells, giving it striated appearances. In addition, aldolase C has been identified as an binding protein in tracheal smooth muscle. However, the implications of this association are unclear. All three isozymes have nearly identical active sites, and all the isozyme-specific residues are located outside the active site. Okay, and as well as the three-dimensional structures of all three of them are similar as well. So we went through a lot of information so far. So let's just quickly look back at glycolysis so we can get a feel for that before we finish up with our last few bits of information. So as you can see here, we have our, let's highlight that, our hexokinase to our phosphoglucomutase to our phosphofructokinase, and seeing how distribution of the use of ATP, conversion to ADP, NAD, conversion of NAD plus to NADH, and in this case, the creation of ATP, one of two ATPs that are created in glycolysis. Let's finish up with aldolase B playing a role in gluconeogenesis and glycolysis, which will cover some of its expression regulation. Thus, any transcription factors that can influence the balance of glycolysis and gluconeogenesis in theory can regulate the expression of aldolase B. So with that being said, they don't directly regulate B expression, but because of their influence on the feedback loop that allow for expression of the protein, and by regulating the pathways, glucolysis and gluconeogenesis, they can then, by proxy, regulate the aldolase B. In a paper by Tolan, it describes how HepG2 cells, HNF1 and DBP, are strong activators of aldolase B, which means the expression will be increased, while HNF3 competitively inhibits HNF1 and non-competitively inhibits DBP. Either way, HNF3 decreases the expression of aldolase B. And lastly, the EPB was found to weakly activate aldolase B in HepG2 cells and strongly activate aldolase B in liver cells found in studied in primary cultures, freshly harvested and exposed to growth media. With that being said, that has been our lesson for today. We've covered a lot of information, but that has been just the tip of the iceberg. There are a lot of different turns and twists that you can explore on this subject matter. And you can actually take many different classes that would explore this, or maybe even make a dissertation of your own about any one of these aspects in further detail. So now feel free to discuss with your partners nearby as we conclude our lesson for today. And as always, email me any questions that arise before tomorrow's class and any other questions before our laboratory investigations. So until next time, I've been Teacher Joseph. Bye.